So I welcome all the students, faculties for attending National Technology Day 11th May, which is proposed by, we have proposed Dr. Professor Nilesh Kumar Sharma from Biotechnology and Bioinformatics Institute, Dr. D.Y. Patil Institute. So he has 15 years of teaching experience and research experience with 105, I think so, sir. I'm not mistaken, na? It's 1.5 publication to his credential, and majority of the publications are in Scopus and Q1 journals. And I really want all of you to get motivated by whatever he talks, because he will be joining the technology with our optometry. So let us listen to him on this National Technology Day, what he has to say and how we can come closer and do something innovative. Thank you, sir. Now I welcome Dr. Professor Nilesh Kumar Sharma to go ahead with the talk.
Yes. Thank you very much, Dr. Nita Madam, and all the students and all faculties. And as she was spelling out, publication itself does not matter. What it matters, the idea and the technology, and how we can help society at large, like patients or anyone. And in that view, what I thought, it's good, opportunity, and also I'm privileged that, on National Technology Day. However, I was aware and we celebrate National Science Day, but National Technology Day, honestly, I'm participating first time. Okay. But yes, because we are looking at the bridging point of science and technology, and for all the student, I think it is also for the knowledge, the government of India has separate ministry of Department of Science and Technology. And you can understand the whole importance of government to give emphasis on science and technology should be worked together. Okay. And that is why it's good day to give, some thoughts and ideas. And definitely 11th May is National Technology Day. So today, if you look at the title, we are going into to discuss an idea how it was converted into a technology, right? Specifically for, if you look at here, early diagnosis of like cancer or inborn error of metabolism. And whole, metabopsy, if you look at the word is here, it is a new word. In Google or anywhere, this word is not in existence. We are the first who has proposed this particular word itself, metabopsy. And we will discuss how this word and what is the whole meaning. And then technology is the VTGE. I will also explain, through the video and GIF, what is the whole importance. And definitely contents we are looking into, it's not like seminar or a routine seminar. So there are certain why, how, what questions are always engaged here. And that will also give a student an opportunity, the importance of 3W. What, how, and why. Always we have to ask, right? Like today we are discussing about the metabopsy or any technology, then question will be asked from the patient, from the student, why we are interested, what benefit we will have, इससे क्या फायदा होगा. That is the many questions are there. So I think I will try to address those questions. Yes, also for a student, I think students are aware. Even I was not much more aware. So this National Technology Day actually to commemorate on 11th May every year for the famous nuclear technology. What should I say? Nuclear technology in the form of Pokhran test or nuclear test. That is the outstanding achievement by the government of India in the recent past, after independence, what I should say. And that is why it is very critical. So nuclear is a technology, right? That's what we understand. Okay. Then Also for a student, it is should be understood very clearly what is like we study every day in the curriculum, semester 1, semester 2. So we study a lot of things, right? In textbook, etc. So what is the whole technology here? Technology we refer as an art or the skill so that we can perform, we can do something. So that is why, technology is nothing. It is an application of what we read through the textbook. We read through some teaching or practical classes. So all the scientific knowledge has to be combined together in the form of an skill or something, so that we can solve the problem of society, like health issues, environmental issues, defense, and many more things are needed. And that is where technology is the key of society for the student, for the country, and each and everyone. And in fact, technology is derived from the Greek word. That means technique, that is art and skill. Okay. So it is not just reading the sentence or reading the textbook. It is implementation, what we have gathered as a knowledge. And that is why students should be very open, not just read the textbook, they should also implement what they are reading the textbook, okay, through their curriculum, like undergraduate or postgraduate. Also, one more keywords is missing, like technique. We also use the word in routine day life. So the technique is slightly different from the technology. So technique, sometimes we refer what we do, what we are doing, for solving certain problems. So the technique essentially is derived from the technology, but technique is slightly different from the technology word. Okay, fine. So if we know the whole importance of technology in science or the society and many more, now we will come to a particular problem. Problem is what? In health for the society, the diagnosis and prognosis is one of the requirement, right? The diseases can be inborn error of metabolism. It can be cancer. It can be diabetes. It can be optometry related eye disorders and many more. So then what? There should be a technology, right, to solve these needs, that is diagnosis and prognosis. So my dear friend, if you go to the development in the technique and technology, there are many technologies are available, like in optometry, technologies are there. In medical sciences, technologies are there for diagnosis and prognosis. So one of the technology what we have added up, bringing from the idea to the technology, that is a metabopsy. If you look at here, okay. So this metabopsy, we will discuss a very small story with certain evidences also, how it is going to solve certain needs of the society, that is in the form of diagnosis and prognosis of human health. Okay. Before, we will go to the technology, it is also for a student, they have to realize whatever technology we discuss, whether optometry or medicine or like metabopsy, what we will discuss today, is not a free of cost. We are not doing charity, So the business model is involved, we sometimes the profit losses are involved here. So that is why if you look at here in the currently in 2022 or even next 10 years, $200 billion is the target of biomarker for, that is for the diagnosis, biomarker based diagnosis or prognosis. It's a huge business oriented area. So that is why any technology, those are going to be developed today, or those technology already developed 20 years back, they have certain contribution in the business. They have contribution in these business sector, that is diagnostic market, prognostic market, and it's quite amazing. It's if you look at the graph here, it's increasing day by day. So that is what also one of the scope. Another very important is metabopsy market. If you look at metabopsy itself, right now it is of around $4 billion. So that means whatever discuss, like today I will discuss, or any other technology what you have, it is also very important to think, to convert idea into the technology is one way, also to think how sustainable the technology will be, how patient oriented will be, how cost effective it will be, whether it is going to sustain the market. That is also one of the critical aspect in idea to technology conversion. Okay. Now I will come to the point. Okay. So if we understand that diagnosis and prognosis of human diseases are important for the society, for all of us. So today I will take two examples of why it is needed, why it is so critical. So I will give you one very simple example. So today, like cancer, we will take an example of human disease and inborn error. If you look at in the left panel, there is a one photograph of, I think, movie from Munna Bhai MBBS, right? So if you look at this patient, who is lying on the bed, he was requesting at least 1 month or 1 week should be given to him by the time he was diagnosed with the cancer, so that he can do a lot of things for his family, for himself. So the time of diagnosis was very critical to him. So likewise, if you look at the perspective of the cancer patients or inborn error of metabolism patients, time is critical. So early, if they are indicated or diagnosed for their human disease condition, that will be great helpful. Not all of us, like those are healthy, they can ignore. Time is not so important, but consider that particular fellow where 1 week or one more additional month for his life was so critical. And that is why the diagnosis, and also the later on, once a disease is diagnosed, prognosis is also very, very critical for the health of human disease patient. And likewise, today I will discuss cancer and one of the inborn error of metabolism. Actually, inborn error of metabolism, I would like to highlight in the context of optometry, that is tears from the eye, how the tears of eye can be used as a source of biomarkers and for the diagnosis. Okay. So this is the patient, it's a time is very critical. 1 month is quite enough for that particular patient. And even look at this particular kid. The kid, their life, their human health, there many more complications are going to come in future. So if the family of those patients are aware, okay, these are going to be probable time frame, at least prevention can be taken, at least certain therapeutics or treatment can be adopted. Okay, that's what we understood why we need diagnosis and prognosis for any human disease condition. Forget about cancer or inborn error. Now, if you think that any idea should be converted into technology or certain means by which society can be benefited, patient can be benefited. So I have created one simplistic model. What I think that for any idea to be converted into technology, like see, idea we think every day, like myself or any student, they think everything every day, like some good idea or the bad idea. But idea has to be converted into the technology. For the first three things are important. One is the definitely facilities, money or the lab. Second is the human resource, and igniting minds are required. Minds are going to push forward those ideas till the level of technology. And third most important thing is required also, that is courage or acceptance. Like if I'm thinking good thing, right, I should have courage. People may say no, people may say yes, people may accept, people may reject. So the courage and acceptance is one of the third key parameters. And fourth, what I think for any technology, perseverance is very important. Do not give up attitude is the critical aspect. If any of us, like myself or anyone, a student, if they can bring together all these 4 or 5 attributes, then I'm sure they can bring up any small ideas to the big technology level. And today I would like to discuss those one of the small ideas, how it is what we are expecting to be converted into the form of technology. Okay. Now, so if you look at the title, what I started in the beginning, metabopsy, the word what we have coined. And look at here. So this metabopsy is a new word, definitely we have coined from our lab. So this whole metabopsy is actually, in a simpler way, it connects to the biopsy, right? Biopsy is a simple, the medical or certain inspection or the examination process from any biological tissues, like it can be tissue, it can be cell, it can be serum, urine, saliva, and many more things. So we have coined by using the same, and OPSY, if you look at here, It is a Latin word that means medical examination or inspection. So we have combined the OPSY, that is OPSY word, with our own metab. That is, actually metab word is derived from the metabolites, and metabolites are the key chemical component of any biological system. Whether it is retinal cell, whether it is liver cell, whether it is any type of the cell, everywhere this chemistry is there. That is the chemical component. So we thought, okay, we should combine together OPSY and metab, and then we created a word that is metabopsy. So today we will discuss this particular technology and area, how what we believe, strongly believe, that yes, it can be converted into a certain patient solving platform. It can be useful to certain patient to an extent, so that the whole benefit of the technology can be translated to the society. Okay. Yes, one more thing, and that's what I will discuss here. If you look at we have also drafted. So this is not this metabopsy word is not for today we have created. It took 3 years time, to think, to understand, to go to the platform. And right now in 2022, if you look at here, surprisingly, we have created a domain of metabopsy. That is nail metabopsy. I will discuss in part. Tooth metabopsy, tear metabopsy, urine metabopsy, saliva metabopsy, serum metabopsy, sweat metabopsy, and definitely the tissue or cell-based metabopsy. So today, for our, for this particular day, I will discuss, emphasize on the nail metabopsy and the tear metabopsy technology. Tear metabopsy in the context of optometry, where the inborn error of metabolism we will touch upon, and one is the nail metabopsy in the context of cancer and inborn error of metabolism. Okay. Great. So all of us question may have why and how the nail metabopsy technology may have relevance for the diagnosis and prognosis. So in that, I will give some evidences and certain illustration of the technology or the method what we have created. Okay. So definitely nail metabopsy, as I said, it is a new thought process beyond serum or urine or saliva, what is routinely used. So we are thinking to go for exploring nail as a source of biomarkers or for the diagnosis and prognosis, right? And the nails we are aware. So nails we are not going to take whole nail, right? So we are going to take only nail clippings. Those are routinely disposed of and discarded by any of us, or patients are healthy, So these nail clippings we are going to take, not whole nail samples, okay? And this nail sample, it's very cheaply available, no cost is involved, and no handling issue, right? Storage issue, no processing issue, and every benefit we have. And definitely this nail as a source of metabopsy, and that is why we have proposed that is nail metabopsy as a tools and technology, okay? Definitely the nail metabopsy is non-invasive, right? No issue of degradation, and definitely it has potential for preventive, diagnostic, and also the prognostic approach today or in future. And it can be also applied to the very even like if the most of the time biopsy tissue from the remote areas, rural areas are difficult to be collected because degradation issues are there, transportation issues are there, many more. But nail sample can be transported even from the remote areas to the place where diagnosis or the testing can be done, and these are the benefits. Definitely, we are one more thing. Any technology, like today I'm discussing metabopsy, any technology cannot be useful until unless the technology should be problem solving technology, right? We have to provide certain solution. So what solution we are providing, that is non-invasive approaches, we have to resolve the issue of invasive approaches, what is going on. Also, the routine biopsy has certain limitations. It cannot be used by each and everyone, right? Expertise are required. Medical experts are required to read those information. But what we are proposing or we are intending, that nail metabopsy should not be so expert-driven technology. It can be understood by even general people, society people, or those are not so expert. They should be able to handle the data and the information so that it should be better understood, easily understood by the patient also. And that is also one of the, you can say, plus point. for the nail metabopsy technology. Okay. Definitely, this nail metabopsy technology, we are not just limiting our to the patient. So this nail metabopsy, we are expanding to the pharmaceutical industries or even for basic R&D, like research purpose. Also for the government agency, forensic applications or police department, we are planning to expand the whole technology for different purposes. Okay, and that's what the whole scope is here. So that's what I said. Any technology should have broader scope that should have applicability. Otherwise, those technologies are not going to be well appreciated by the society or the government. Okay, here. So whatever metabopsy or nail metabopsy I was discussing, here is the GIF. If you look at here, so this GIF is true sense GIF. It is really recorded during the experimentation in our lab, in our cancer and translational research lab at the Dr. D.Y. Patil Biotechnology and Bioinformatics Research Institute. So this is the real experimental or the technology we have developed. It is also submitted to the, deposited into the YouTube and many more social networking sites. And here is the actual technology model in the left panel. If you look at, also you can look at here, this is small technology or the idea we have progressed. That means idea that was conceptualized 3 years back in 2019, I remember clearly. From 3 years, the whole idea is expanded, right? In 22, even it is going to further expand. And that is what the whole potential of any technology. Technology also should not be like that, once technology is developed, the whole scope is limited. Whole scope is redundant. Those technology cannot survive in current competitive environment. Okay, so any technology can survive, those can be evolved in time frame. So I remember idea was started in 2019. Even COVID-19 period was intermittent, but still our ideas, we never gave it up. And in 2022, if you look at many more publication, many more, the scope we have created, including with Dr. Nita Madam, we are exploring or we are challenging the inborn error of metabolism by tear metabopsy. Okay, so this is the whole technology. And if you look at it, it's so inexpensive. It is not so expensive, right? It is not like 5 lakhs or 10 lakhs of rupees. It is of 1,000 rupees or 2,000 rupees. We can create we have already not create, we have created. It is already working. So that is what good thing. So we refer this technology as a VTGE technology, vertical tube gel electrophoresis technology. Okay, it's very simple. It looks simple, but idea what we believe is very strong in a scientific manner. And that is why from 19, even the COVID-19 was there, we kept it on pursuing, right? And now in 2022, platform is open. We can do more work. We can engage with many other doctors, hospitals, and many more. And that is why from 19 to 22, I see very better prospects, in the field of diagnosis and prognosis. Okay, I'm not going to go into the details of the scientific model of this particular VTG, but what I can assure you, it is very sound based on the biotechnology, you can say some basics, scientific basis, and many more things. That is why it is sustained, right? It is not only sustained in the laboratory, also by the peers, external experts, or the reviewers. They looked into the methods, they investigated, and it looks very sound and robust. Okay, fine. So I'm not going into the detail, but yes, you can go into the YouTube and other channel where the whole videos are uploaded. This is just a glimpse of the VTG technology. Okay. If you look at here in a very simple way, the left panel, here the samples are going to be loaded, like nail sample or the tear sample or the serum sample or the urine sample. In the left panel, you can see the arrow here. At this place, the samples are going to be actually loaded. And in the right panel, if you look at here, in the right panel, can you see the electrodes? And there is a Falcon's tube, plastic tubes are there. Here is the actually biological samples are going to be loaded. Okay. Now, another question comes to my mind and all of us. Okay, what if, whether this technology is completely new on the earth, whether technology is 100% new? No,
If you look at in the right panel of this slide, so there are some equipments are there, images are there. So these equipments are in existence, like in USA, and there are many US-based company, they create this equipment. Look at the complexities. It's so expensive. It looks, image, you can see it's so expensive. And our equipment, if you look at here in the left panel, it's very simple, it's one tube is only there. But the whole application is always competitive. And if you look at the cost of the equipment is 1 by 100, means 1% of the cost of this existing equipment, our equipment. It's cost effective, huge cost effective. Another important issue is that is the operational expenses is also very critical. Any equipment, they need operational expenses. Chemicals, reagents, tear and wears, some parts are going to be broken time to time, when we use the equipment. So here 1 by 20th, it's very negligible operational expenses are there. So that's why it also gives us freedom to go for a little bit less expensive diagnostic, in future, if we are planning, right? Another very important, this particular design of the equipment, if you look at here in the left panel, is one of the unique. In Google, there is no such images right now. Only images, if it is there, only from our institution or the lab. There is no such images. At least we have gone through. That is what the uniqueness. Definitely, all these benefits, what we believe that even in next 10 years, we have the better scope and we can be competitive. Another important I would like to discuss, that is benefit. User friendly. Like, I can ask 10th class student by giving training of 1 hour, they can do the job. I do not need like a PhD or whatever. I mean, they can do better job, but I can train, that is what the user friendly. Like if you go to the routine biopsy, so technologies are needed, right? Another very good thing is that the unique idea. The idea behind this VTGE metabopsy or the Nail Metabopsy, a very strong scientific basis. And that is why the instrument, what we are expecting, it is going to serve the purpose of diagnosis and prognosis. Okay, so these are the certain advantages, my dear friend. Now here is the data. Okay, we have discussed the technology like nail metabopsy. Okay, nail clippings we are going to use. There are some equipments we are using. How the data is going to be presented for the patient, for the outside. No, those are not expert in biotechnology, or those are not expert in whatever this particular field. So here is the simple explanation. I will try my best. Okay, look at here. So this is the form of the data is going to be generated from the nail metabopsy. And here in the right panel, if you look at here, look at, so we go for the routine blood test in the diagnostic lab. So what we get the data we get, okay, here is a normal range, like 150 milligram per deciliter. And if our blood sugar level is 180 milligram, then they give the idea, okay, your blood sugar is high, or, you have some issues. So likewise, our nail metabopsy, we are planning to provide the data to the patient in the same manner. Like for an example, if you look at Alzheimer patients are there, okay? So the Alzheimer patient is not aware 100% whether Alzheimer's symptoms are there, yes or no. So for the Alzheimer patient, one biomarker that is 5-HL, hydroxylysine, it is a technical term, right? So what I'm giving you the idea how the data is going to be given to the patient through the metabopsy. So we are planning, okay, so suppose for any healthy individual, if the level of 5-HL is below 100 AU per milligram of the nail sample, as I said, we are utilizing the nail clippings, right? So we know the amount of nail clippings. So per milligram of the nail, this is the healthy range, right? If in a particular patient, if the range is 2500 AU per milligram of the nail, then we can suggest that you are at the early stage of the Alzheimer or those neurodegenerative, and then you should take preventive care. So it's very simple. So the data is not going to be presented to the patient in a complicated manner. Data has to be presented to the simplified manner. And that is what the technology. Any technology should be user-friendly or the patient-friendly. And now you can see the graph here. Look at here. So like patient 1, patient 2, just for an example, the patient 2 has the symptom of or the level of 5-hydroxylysine, that is a biomarker of Alzheimer patients. And in this graph, surprisingly, if you can notice very clearly, there is a doxorubicin anti-cancer drug because one of the patient was cancer patient. Look at here. So in the nail sample, even the anti-cancer drugs are deposited. One more thing, there is one tumor biomarkers, that is deoxyribonucleic. So it's very surprising to all of us. Huge amount of information or the data is in the nail metabopsy. The challenges are in front of us, how the data is going to be given to the patients so that patient can get benefited as early as possible. You know, the time anyone will be late, they will be loser, right? At the loss. If they are a little bit early, then they can be benefited. That is what the whole presentation of the data from the nail metabopsy. Okay. Another example I would like to show here. So this particular slide is more scientific. So how the machine is going to generate data through the the process and the protocol. So this is the actual data generated by the nail metabopsy approaches. And if you look at here, this is one of the example of oral cancer patients where the nail clippings of oral cancer patients are utilized as a part of nail metabopsy. So my dear friend, what I'm going to give you very brief idea that the nail metabopsy, whether for oral cancer or the breast cancer, the scope is there. Only it is a matter of how we are going to interpret, how we are going to translate the data into the patient friendly manner so that patient can understand and then they can be benefited for early diagnosis. Now, my dear friend, one more interesting thing I would like to show here. During the nail metabopsy, we have also surprisingly found certain biomarkers for the diagnosis of the inborn error of metabolism. So look at here. In one of the patients, these are the biomarker of inborn error of metabolism. Those are deposited in the nails. Even the patient is not aware that patient is the carrier of inborn error of metabolism. Sometimes what happens, inborn error of metabolism, they may prop up at certain age, right? They may create complication due to certain diet, but even patient is not aware. So look at here. So these are the whole profile of, surprisingly in the nail metabopsy of patient, these inborn error of metabolites are detected. So definitely, it can be presented to the patient and patient can be benefited from those ideas. Okay, now I will come to the section of optometry. That is of inborn error of metabolism for the tear metabopsy. So my dear friend, if you look at here, the tear metabopsy, as I said, for any technology in the diagnosis, the critical aspect is the tear, like biological sample collection, right? How we are going to collect, how better we can do, right? So this is the first step. Second step, the samples are going to be processed, purified. That is what the technology I have mentioned earlier, right? And then third aspect is the analysis. So all the three steps we have in our hand, we have developed our own methods. And that is why if you look at here in tear metabopsy, what I can predict, tear metabopsy can touch upon or can help, like definitely eye disorders like diseases for optometry, like glaucoma or diabetic retinopathy, inborn error, and then cancer can also be touched upon by this tear in future, not today, right? Now, if you look at here is the method for a student, what I understand, at least they can appreciate in the perspective of optometry. That's why I created a process. Here is the, if the tear drops are there, those are going to be collected by the strips. You may be aware, you guys. And then collection of the tears will be directly submitted to the tube where the patented metabopsy elution buffer we have. It will be transferred here. The time it will be transferred, the whole tear samples will be collected here in the buffer, and then it will be loaded on our own VTGE tools as I have described earlier. So it's very simple. Only thing is that how the data, once it is generated, it is going to be understood by the clinical doctors, how it is going to be understood by the patient, and how it is going to be beneficial to the patient. That is a critical aspect. So we have the technology, but yes, translation into the reality, definitely it will take, and Dr. Nita and I think with medical college, we are working upon it. Let us see how it turns out, okay? But what I can assure, it will work because the method and process are very sound and we have certain evidences, right, in our hand. Another aspect is, the beyond inborn error of metabolism. I have gone through that in optometric tears. What I think is a mirror of healthiness of any eye system, eye visualization. So tears have various chemicals, right? Various protein, various things are there. And for a student, if they may be aware, tear, where in the eye part, the most of the cells are immune privileged. Immune privileged systems are there. There is no immune cells as such, for rejection or anything. It's a very different type of the organ system. So the tears, those are generated in those areas, they may have very exclusive nature of certain chemicals, certain protein. That is why tear metabopsy are very important. But how to get the data out of tear can create a fear, okay? That is why the technology has to come, to remove the fear from the clinical doctor, from the patients, whether it can be translated into a beneficial thing, for the patient as well. Definitely, for all this metabopsy, those they can also help out, right? So we have certain various social networking, YouTube, we have our own website, Twitters, everywhere we are sensitizing the whole technology and the methods, so that not today, maybe in future, the more sensitization, awareness, so that patients and clinical doctors will come forward, right? Not only clinical doctors, even the municipal corporation, police department, we see the prospects to work together, okay? And already we have discussed the different domain like serum or urine or nail, etc. So it will take 50 years, 20 years. I do not know how many years, but yes, technology, it takes time, right? Yeah. So the last one, that, as I said, the journey we started from 2019, right? Before COVID-19, yes. And in 3 years, we have created huge progress, right? You can see the whole domain we have created, word we have created, terminology we have created. So at least 6 to 8 patents and few more are in pending, more than 20 Scopus WS indexed papers are published. More than, I think I can count, if I can count, more than 20 undergraduate, 8 PG, PhD students are trained in one simple idea or the technology. So you can understand how the idea was expanded from very beginning to the long way, like in 2022. And 2022 is not going to be the end of the day. I hope that it will also expand. And also we have created, that startup company, it is created a startup company as a Nail Metabopsy Health Services Private Limited through the DPU, okay? So what I think, it's so great. At least we are so motivated, and I hope that the students will be also motivated. So definitely at the last, to acknowledge our Honorable Chancellor, Sir, Honorable Pro-Chancellor, Madam, Honorable Vice-Chancellor, Sir, Honorable Trustee, Madam, and specifically Professor J.K. Pal, Sir, head of the institution, who has definitely motivated for behind this idea. And he never said no, that this cannot work, he promoted. And also the DPU Incubation Center, Central Research Facility, all other resources they have contributed for the data generation, right? And definitely undergraduate student, and postgraduate and PhD, all they have. See, idea was one sentence, idea was from my side, one sentence in 2019. Now the idea is so expanded in various form, either publication or patent or a startup company. I feel so amazed and pleased, to see the word or the idea is expanding, okay? And the last, you can see the, here is the laboratory. I lead this particular lab in the Biotechnology Institute. It's a big room, separate room is there. Every facilities are there, it's, yeah. So these are the students. So it's whole, this is referred as Cancer and Translational Research Lab. It's a dedicated lab, right? So all the data is generated there, like all the equipments are there, VTGE equipment is there, everything is there, right? So all the students they work, Professor J.K. Pal, you can see myself, Professor Srode, and many more like now Dr. Nita Madam, we are, and next time once the presentation will be next year, I hope so, Dr. photographs of Dr. Nita Madam will be as a collaborator, I believe, and I assure. And a whole journey, it's credited to the student, no? Otherwise, if I say idea is here, no one is going to buy or purchase or accept, it does not matter, right? So these students are also acknowledged so beautifully. And yes, and the last is kind attention. And if any question, that will be great. Yeah, otherwise.
Any student, anybody wants to ask any question from sir? Or any faculty for that matter? Sir has given really a very good talk on Nail Metabopsy, and surely it's an innovative thing. And as you said, a lot of early detection of anything like cancer, Alzheimer's with the nail. Then we have this retinoblastoma, which is a cancer of the eye. So those things can also be detected and taken care of. Anybody, nobody is asking anything. Everybody knows everything.
I think, yeah, they have understood, or they might not have understood. So it's like a positive and negative, right? 0 and 1 code.
Yeah.
But it's okay. At least, what I believe, if 1% of the student,
Yes, sir.
at least they might have understood the whole definition of technology, the whhole idea. In
Yes,
their
sir.
optometry field, definitely we are working in the biotechnology area, right?
Yes, sir.
So if at least one student, they will be motivated, technology driven,
Correct.
that will be great success of National Technology Day, what I feel.
Yes, sir.
Otherwise, I know, yeah, it needs a lot of courage and mind to think into that direction. Okay, good.
Yes. And
So,
I request all the students and faculties to please switch on your camera so that I can take a photograph. All of you, please.
I should unshare, right? Then I think you can.
Okay, sir. I request all the students and faculties to switch on your camera. Five count. 1, 2, 3, 4, 5. Gaurav, where is your camera? Please switch on, all of you. Good. Take it. So, smile. Did you get anything from, were you listening to sir? Say yes. Open your mic and say yes. Say yes.
Actually, they might have understood. They will implement later on.
Yeah.
And yeah, so they will implement.
No, sir, nobody has written thank you, sir. Kam se kam bolo, muh se.
So I'm sure that they will implement today or in future.
101%, sir. Yes. Okay. So I'm taking a photograph. Please smile, everybody, to get a nice picture of yours.
You can, madam, go for a window print screen if it is laptop, so it will capture.
Sir, वो try किया हुआ है.
No, it does not work.
अच्छा करते हैं फिर से.
Window print screen you can show.
ये है ना print screen. ये print screen.
Word document open करो.
ठीक है. Okay.
Good. Thank you. I appreciate optometry student. They didn't ask, a question, and it also understood in this way, so that they are in the mode of implementation. I think from today they will start working, in their optometry field to design or to innovate something.
Yes, sir.
Okay. And once it is innovated and designed something, please also inform us that at least the message has been transmitted.
Yes, sir.
To the student. Okay. Thank you. I think we should.
I thank you, Nilesh sir.
Thank you very much.
For listening to my request and just saying yes in one go itself, without showing any type of attitude or anything like, okay, I have done such a big invention and why should I come to optometry and talk about it? So
no, I have not come to the
optometry. Down to earth attitude. Your down to earth attitude, okay, and the willingness to help and to talk about your technology to everybody, and to share it with our students. It was really great, and hope next year, as you said, surely we'll come out with something to talk about this year.
Even I learned today that National Technology Day, other than National Science Day, what we used to celebrate. So I knew additional thing, right? Yes. So now I can feel proud that what we are working, maybe today or tomorrow, connected with the mission of national level, right? National technology, right? So it's great. Yes. Thank you very much, and best of luck to all of you, to you and all your department.
Thank you so much.
Thank you, madam. Bye bye.
And thank you all the students for joining. Okay.